Hello, class. This is your professor, and this is chapter 10. Chapter 10, we're talking about patterns of inheritance. So, we've talked about genetics, chapters 7, chapters 8. We talked about DNA, we've talked about genes, we've talked about proteins, we talked about chromosomes. So, now we're talking about how characteristics are uh, passed to generations, to the next generation, and what the DNA says, and then also what we actually see. So we're going to talk about genotype versus phenotype. So a gene is a segment of DNA, as we talked about. DNA is you have quite a bit of information in DNA, but a gene is a segment of that DNA that carries the instructions for synthesizing a specific protein. So a segment of DNA that will code for a specific protein is called a gene. Those proteins could make different things, skin color, size, shapes, um, different parts of the body. Proteins are sort of like the building blocks of the body. And so we look at the study of genetics, gene, see the word gene in genetics. We look at the study of genetics to study how genes are transferred from parent to child and how genes are expressed in the offspring. Right. So chromosomes, as we've mentioned earlier, before in the previous chapters, where chromosomes are um, pieces of chromosome is a piece of DNA containing many different genes. So on each chromosome, you have pieces of DNA. So each chromosome is a piece of DNA, and on each chromosome you have different genes. Each gene, the location of each gene on that chromosome is called a locus, right? So just breaking down, helping to understand how we divide it, how we look at it. Haploid sex cells. Remember, your sex cells are your sperm and your egg. Sperm and egg have haploid, half the total number of chromosomes. Haploid sex cells fuse during fertilization. The sperm comes and fertilizes the egg. And now you have half the DNA from mom, half the DNA from dad, comes together to make a diploid zygote. Zygote is a fertilized egg that has a full set of chromosomes. Each chromosome, which is a segment of DNA, part of your DNA, is a homologous pair. Humans have 22 homologous pairs of autosomes and one pair of sex chromosomes, right? Total of 23. And 23 pairs equals 46 chromosomes. Diploid cells have two alleles for each gene. Okay, so members of the homologous pair have the same genes, but may have different versions, right? So we're going to talk about versions of genes. So some genes may express, like for instance, you may have the gene for your eye color, and you may have a gene that expresses blue eye color, but you may have another gene that expresses no eye color, or let's say brown eye color. And so you may have two, they're in the same location. They are homologous pairs at the same location on each chromosome. Each pair of chromosomes, they're at the same location, but they are different versions of the same gene. That is what we call alleles. Alleles. Here are some questions you can ask yourself. Uh, how does things like meiosis and all these things play into gene expression? Right. So how did we discover that DNA was the way that genetic information was shared from one generation to the next? Well, Gregor Mendel, scientist, which you are a scientist because you are studying science. But Gregor Mendel is a scientist. He used the pea plant. Um, Plants are very easy to control, and he was able to pollinate, which is sort of like how plants have sex, but able to pollinate one plant with another. Not really sex, but they can pollinate. Therefore, one can breed with another. And then he planted the seeds and saw what happened. Right? From green plants, um, true breeding plants produce offspring identical to themselves. So certain plants produce offspring when plants were fertilized themselves, they produce the same color uh, offspring, same color seeds as you see here. But when you had one plant fertilizing another plant, so you were crossbreeding the plants, you saw a the mixture of the seeds. Some alleles are dominant, and that's what he discovered in his research. Dominant alleles exert their effects whenever they are present. Crossing a yellow seed plant with a green seed plant always yields some yellow seeds. Therefore, the yellow seed color is dominant, whereas the green color is considered recessive, right? Recessive. And so that's what we're going to talk about. Recessive alleles usually encode something that is not functional or not as functional, right? So it gets dominated by the other allele. So why do some plants produce yellow and some produce green and some produce yellow and green? And the answer has to do with the two alleles for each gene because of the homologous pairs of chromosomes. Right? So we have two ways you can get a yellow plant, but only one way you can get a green plant. So this is where we have the term phenotype and genotype. Genotype is the actual DNA, whereas phenotype is what we actually see, right? So if you look at the chart here, the genotype homozygous dominant, that means the two alleles, so on two locations of the chromosome, the two locations of the chromosome, you have the two alleles for this color gene. If mom gives me a dominant Y and dad gives me a dominant Y, then the child will be dominant Y, yellow. Mom gives me a dominant Y and dad gives me a recessive Y, a different version, has a different allele, then because of the dominance of the yellow, of the capital Y, the dominance of the yellow allele, the seed will still be yellow. But if mom and dad both give recessive Ys, we designate that with the lowercase y, then the child will be, the seed will be green. Right? So it just depends on what is coming from mom, what is coming from dad, and which alleles we are getting. And based on the genotype, we can figure out what the phenotype will be. And so we looked at the offspring to determine the genotype of the parent. So based on what we get, we can also determine what the parent must have been, because the parent can only give one allele. Right? And so that's just talking more about that. The seed color gene encodes a pigment metabolizing enzyme, which is a protein. The dominant allele Y is active and produces yellow color. The recessive allele, lowercase y, that's how we designate that, is inactive, and the plant would stay green. Right? So two recessives give you green, two dominants give you yellow. A dominant and a recessive will still give you yellow, but maybe some green depending on who you mix it with. And so here we have what is called a Punnett square. Uses the genotypes of the parents to reveal which alleles the offspring may inherit. And so in this case, In this first case, if we have mom is yellow and mom is heterozygous. Heterozygous means not the same. So mom has a dominant Y on one of her alleles and a recessive Y on the other. So her phenotype is yellow, but her genotype is heterozygous, and she mates with dad, who is a recessive, who's dad who is also heterozygous. Heterozygous. So capital Y and lowercase y, also heterozygous. Then the chances of offspring being yellow are 75%, as you see in the square. You can end up with the two capital Y's, two heterozygotes, and one recessive, based on the combinations that could potentially come out of this. And so the likelihood of two heterozygous parents having a heterozygous yellow parents having a recessive child is 25%, 1 out of 4, right? And this is called a monohybrid cross, one trait monohybrid cross, since both parents are heterozygous. All right, and so it's just talking more about that. When the cells divide by meiosis, meiosis is how we form sex cells. Each sex cell, each sperm, in human beings it would be each sperm and each egg, but each gamete, those are called gametes, would receive one allele. Remember, they have half the number of DNA, so they're either going to get so they would either receive a capital Y or a lowercase y if the parent was heterozygous. So that gives us just more showing us what that would equal out to. Right? And the chances of the green is 25%. So Punnett square helps us to answer those questions. If the yellow seed color is dominant, why are some seeds green when a yellow seed plant is crossed with a green seed plant? All right, so the Punnett square can help us answer those questions. Right, so if a cross between a yellow seed plant and a green seed plant yields all yellow seeds, then that would tell us that one of the parents is, the yellow seeded parent is homozygous dominant. They have two capital Y's in this case. But if a cross between a yellow seed plant and a green seed plant yields some green seeds, then that tells us that the parent is heterozygous, because the parent must have passed on a lowercase y. So Punnett squares can summarize meiosis fertilization. So two alleles for the seed color gene are packaged into separate gametes, then combined at random. And Mendel's law can be applied to human traits, right? So it can be useful for tracking disorders, such as, in this case, cystic fibrosis, which is a very deadly disease, but two people who are healthy could still be carriers. In other words, they're heterozygous, could still be carriers for the disease. And so if two people who are heterozygous for this particular disease were to come together and have a child, there's a 25% chance that their child could have the disease, and then there's a 50% chance that their child would be at least a carrier for the disease. Right? You can also look at dihybrid crosses. So we can potentially track two traits on different chromosomes, right? So we have two different traits that are on two different chromosomes, and what are the chances that due to the combinations that could potentially, what are the chances of all the different combinations that could happen, right? And so you can see here that Mendel proposed that there's a law of independent assortment. So if I have two traits on two separate chromosomes, there, and mom gets half, each gamete gets half of mom's, each each egg has half of mom's chromosomes, each sperm has half of dad's chromosomes, and so we have they're going to randomly assort, right? And so we can, they in this case, if they're on two different chromosomes, two traits, so we have maybe eye color on one chromosome, we have height on another chromosome, and let's just say, and those traits, you know, on a chromosome means they're on two alleles for eye color, two alleles for height, just for an example. They would independently assort. So you would have, some people would have blue eye color and be short. Some people would have brown eye color and be tall. Some people would have blue eye color and be tall. You know, so depending, it would independently assort. But genes close together on the same chromosome, so for instance, if we have genes that are all on the same chromosome, well then if you get, when those chromosomes, when those homologous pairs split, they would be linked together. So Punnett squares and the product rule assume two genes are sorting independently. So when genes are linked, they are physically near each other, therefore they do not sort independently, and linked genes are not inherited independently of each other. So there would be, you wouldn't be able to just do the basic Punnett square for independent. The original Punnett square assumes independent assortment, but if the genes are linked together, then you would have more. You wouldn't have the variability that you would have if they were independently assorting. And then we also have figured out that not all genes follow the same inheritance patterns. Right? So we've generally discussed, when we're talking about the green seed and the yellow seed. There was green, there was yellow. There was no in between. Sometimes you have alleles. So alleles, two locations of the gene on homologous pairs. You have the alleles, and you don't have the dominance of one over the other. Dominant alleles mask recessive alleles, but sometimes you have alleles where the phenotype, what we see, is a blend of the two. Incomplete dominance is what we call that, right? So it has an intermediate phenotype. You have one gene codes for red, one codes for white, and some of the offspring in that case, some of the offspring come out pink in between. Incomplete dominance create a blended heterozygote phenotype. So in this case, red allele R1, white allele R2 are both present, and you have this R1, R2 phenotype is this intermediate pink. You also have codominance, right? So incomplete dominance is, you know, we're both dominant, but we blend together. Codominance is different than incomplete dominance because codominance says I'm dominant and you're dominant, and we're both expressed independently. Now, you would have this if this were in the previous example, you would have the red flowers and the white flowers, and let's say you had a flower that had some red and some white all on the same plant. That would be codominance, but you don't. In that case, you had pink flowers, which is incomplete dominance. But in codominance, as in the example given here is blood type, you have on the surface of your blood cells, red blood cells are called erythrocytes. On the surface of the red blood cells, you have antigens that our bodies code for, and that's what gives us our blood type. So type A blood, type B blood, type AB blood, type O blood has no antigens. All right, so when you can have type A blood, you can have type B blood, you can also have AB blood, which has A antigens and B antigens. They're not blended together, they're both existing at the same time on the surface of your blood cells. And that's what it's talking about here. Then you can also have type O blood, which has neither one dominant. Doesn't that's the recessive version where you don't have A or B. We also have what is called pleiotropy, which one gene has multiple effects on a phenotype. For example, a gene might affect more than one biochemical pathway. So in this case, Marfan syndrome can affect your eyes, it can affect your bones, your joints, your heart, and your lungs. So one gene having multiple effects, pleiotropy. You also have epistasis, occurs when one gene's product affects the expression of another gene. And so back to the type A blood or type B, you have to have a way for that antigen, that A antigen, to be able to bind to the red blood cell. So the expression of the H allele, gene 1, affects the expression of A and B alleles, gene 2. So one gene affects the other genes because you have to have one, the H allele, to be able to connect the antigen to the red blood cell. So those genes, if I didn't have the H, I couldn't, even if I had the A couldn't connect to the red blood cell. So epistasis, one gene affecting the expression of another gene. You also have sex-linked genes, right? So genes that are on the X chromosome, what we call the X chromosome, right? So in humans, females have 2 X chromosomes, males have 1 X chromosome, 1 Y chromosome. And a Punnett square shows that each fertilization event has a 50% chance of producing a female and 50% chance of producing a male. If mom can give an X, dad can give an X or a Y, then there's a 50% chance of the children being female, 50% chance of the children being male due to the Punnett square. So the egg will always carry an X chromosome because mom can only give 2 X's, only has 2 X's, so the egg will always have an X chromosome. And so really, it's the male, it's the sperm that determines if the offspring is male or female. If the sperm is carrying the father's X, then the child will be female. If the sperm is carrying the father's Y, the child will be male. Another thing that's interesting is when genes, remember genes are specific locations on the DNA that code for a specific protein. When genes are on the X chromosome, males only have 1 X chromosome. So whichever allele males have on their single X chromosome is the one that is expressed as a phenotype because they only have 1. All right, and the example here talks about eye color. Right, so if in this case, you see the male has a dominant R on the X chromosome, so they'll have the red eyes and only be able to create females with red eyes, versus if there is a male, it could have red eyes or white eyes, because their chromosome, their allele, their gene is only carried on the X, not on the Y. So that's what we see when we do the Punnett square. Females have two alleles for genes on the X chromosome, so it gives them more variability in what they can transfer into the next generation, as you see here with the Punnett square. X-linked recessive disorders affect more males than females, right? So males only need to inherit one X-linked recessive allele to express the recessive disorder. Females must receive both of the recessive allele, right? Which makes sense. If the male is dominant, the female in this case has to have the male. Well, the male will have more likely will have the trait, because the father can only give the dominant a XH or a Y, and the female. So the only chances of having a child recessive to hemophilia in this case is if it's a male. The female can't make the mother cannot make a female with the disease. And this is a chart that talks about some sex-linked genes. Disorders can be dominant or recessive, and some of the ways those things can happen. You can look through this chart. We also have what is called X inactivation, prevents double dosing of gene products. Each cell in an XX individual, such as these female cats, randomly inactivates one X chromosome. If one X chromosome has an allele for orange fur and the other has an allele for black fur, color patterns emerge when X chromosomes are randomly inactivated. Sometimes this happens, randomly inactivated, X chromosome inactivated. In humans, an X-linked, X-linked dominant disorders are less severe in females, which makes sense. In humans, because females get two, get one from mom, one from dad. But in humans, an X-linked disorder called Rett syndrome is lethal to boys and has varying effects on girls depending on how many cells inactivate the X chromosome carrying the Rett allele. So the X can be inactivated. There's a question about that. The environment can also alter your phenotype. So many genes are affected by the environment, and that's interesting. But think about it, DNA is a molecule. DNA is a substance. DNA is not it's not a computer program. It's a molecule. It's a living it's a living thing. It can replicate. It exists inside the nucleus of your cells, so it can be affected. Things what the DNA does, coding for proteins, and those proteins can be affected by the environment. So many genes are affected by the environment. For example, the enzyme responsible, remember we say the enzyme is basically a protein. It's a specialized protein. The enzyme responsible for pigment production. Remember, an enzyme is also a catalyst that helps speed up the reaction by reducing the activation energy without being consumed by the reaction. The enzyme is responsible for the pigment production in Siamese cat fur. and it's active only in cool body parts. In warm body parts, no color is produced. See this? So the gene is acting in certain body parts. Remember, proteins can be affected by temperature. Some traits depend on multiple genes, right? So just like we have some genes that can affect multiple traits, we have some traits that can affect multiple genes. Skin color is a polygenic trait. Poly, many, genes, polygenic trait. It is affected by more than one gene. So multiple genes help to make up skin color. And isn't it beautiful seeing all the different colors of skin? It's a beautiful thing.